Thank you all for being here. I have 10 minutes to give kind of an overview of determining, using experimental methods, the function of an SCN2A variant. I won't be showing individual variant results. It's not possible in 10 minutes, but I want to give a framework for how we do this and why we do it. I think already we've heard from other talks why we do it. We want to understand, first of all, is this variant capable of disrupting the function of SCN2A in a manner consistent with the condition of the patient? That's number one, establishing pathogenicity. It may also give us a clue, as you've heard, whether it's polarized functionally toward the gain of function spectrum or toward the loss of function spectrum. And that can help us also understand potentially whether an individual will respond to certain drugs or not. There are additional functional subsets other than pure gain and loss that we also can reveal when we do this kind of analysis. And I think in the long run, ultimately, as new therapies are developed, those therapies are going to be highly tailored to variants that are either gain or loss of function. And I think that's another very important reason to understand. Now, it's not possible to do an experiment on every discovered SCN2A variant. There are just too many of them. But the data we've generated can be used to help build computational models, and you're going to hear more about that in a later talk. Just for big picture, SCN2A is a sodium channel. It's a gated sodium channel. Its activity is modulated by the voltage in the cell and in the cell's membrane. And when a neuron in the brain is not firing, sodium channels are closed. They're not active at all. But then, as soon as the neuron is stimulated to fire an electrical impulse called an action potential, sodium channels pop open and go into action. In fact, these are the major drivers of action potentials in neurons. They don't stay open very long. They shut down very quickly in what's called inactivation, and then they'll stay closed in that state until the membrane recycles until it resets itself. So this is the paradigm that we think of when we're trying to analyze the functional properties of a sodium channel. How does it activate? How does it inactivate? And are there properties even beyond those qualities that we can ascertain? So the classic way of assessing that is using this neuroscience method called patch clamp recording or patch clamp electrophysiology. And that can be done either the traditional way, as shown on the left, with instruments and expert individuals who are trained to do this, or one can use a now available automated method, which is what we've really embraced recently. And just to give a nutshell, when we're talking about sodium channel function, when sodium channels open, we get this big deflection of recording of current in the cell. And we can measure several parameters there, some of which you may have heard about. We can identify the peak of the current, what's the maximum amount of current conducted by that sodium channel. We can look at the size of the cell and then equate the number of sodium channels that must be firing into what we call current density. This is a standardized measure. And if you have more or less current density, you can start to interpret that as gain or loss. If there's no current density, clearly a loss of function. If there's a massive amount of current density, which is not that common, we could call that a gain of function. There are also some really nuanced properties. If the channels don't completely close after they open, you might have a small amount of residual current that we call persistent current that has also been identified in many SCN2A variants that we would label as gain of function. This shouldn't be there. This is an abnormal property. Now, that's just the very beginning. There's many more properties that get measured, and I don't expect anybody in the audience who hasn't been around electrophysiologists to understand any of this, but there's a lot of functionality that can be ascertained from a single experiment on sodium channels. And ultimately, we try to distill all of that information down into this very oversimplified, can't emphasize that enough, oversimplified spectrum from gain to loss of function. And the association with the diseases has been somewhat clear-cut, that early onset, neonatal period onset epilepsy is often associated with the gain of function mutations. Later onset neurodevelopmental disorders with or without seizures is more loss of function. And so that distinction is how we try to simplify everything. But there are many variants that have mixed properties that are difficult to classify into one of those two buckets. And that's where the field is right now. There are more advanced neurophysiological techniques to try to address this. They're not available for a high throughput strategy yet, but that's where we are. That's where we're moving. So try to getting away from this and trying to develop a more nuanced approach. Again, there are lots of SCN2A variants, most of which have not been studied functionally. This drawing here simply represents the nearly 900 variants that have been just reported in the scientific literature. In the ClinVar database, which is a repository for genetic discoveries in all genes, there's over 2,000 SCN2A variants. And in that group, there's probably over at least 1,000, maybe over 1,200 that could potentially have some functional consequence that could be determined experimentally. Unfortunately, the large majority of those variants are of uncertain significance. We don't know how to interpret them. Are they even the cause of the disease? And certainly, it's difficult to look at them and interpret them as gain or loss of function in many cases. Now, one area that's near and dear to the family's SCN2A Foundation is their clinical trial readiness study. And in collaboration with Ann Berg and others involved in that study, we've made it a point that we will functionally test every variant in that study. And I think we've done that to date. This simply is a rough summary of how many of those variants are loss of function, LOF, versus gain of function, GOF, or have mixed features. You can see there's no pattern here. It's really impossible to predict that from just simply where the variants are located. But that's progressing very nicely. And this is just sort of an example of the volume of data that comes from this kind of work. The last thing I just want to finish with is these are experiments that are done not in neurons. They're done in another kind of cell that allows easy manipulation in the laboratory. And there are some definite advantages to using these cells and this approach. There are also limitations. So the key advantages are you can make very, very precise measurements in high throughput fashion using this approach. You can really understand subtle nuances of function, not just gain and loss, but get into more of the detailed channel behavior that's maybe present. And what we've noticed is that mutations don't all fall into the same bucket. They're all very different from one another. These systems experimentally can also be used to test drugs and test the effect of drugs on individual variants. And ultimately, one can use this to guide which variants might be most interesting to move forward to things that are much more expensive, like making a genetic mouse. The limitations are shown here on the right. It is slow. It doesn't happen overnight. It is not a clinical test. You can't order this at a hospital. And it's really limited to specialized groups who've developed the methodology like ours. Now, we're going to hear later in this session about predicting variants, and I won't belay that, but one could also make the same arguments of advantages and limitations. I think these are going to come up later. The major advantages, you can make a prediction right away. You don't need to wait for an experiment to be done. These are systems that have been trained based on experimental results from elsewhere, but it's ready to go. Whether that's accurate enough for determining whether someone should be in a clinical trial or a gene therapy is to be determined. So anyway, I think with that, I'll just share some acknowledgments of my group and our funding sources. This work has largely been funded by our soon-to-be ending Center Without Walls from the NIH and some former funding from the Safari Foundation. So thank you very much, and I look forward to the panel discussion.
Thanks, Al. That was fantastic. I'd like to welcome up our next speaker, who is Dennis Lal. Dennis has achieved things with computers which many of us can only dream of. Today, we're going to witness a miracle where Dennis claims to be able to get through 53 slides in 10 minutes.
Okay, let's directly jump in. So, it's limited time, right? So, you know, if there's anyone in this room who thinks this is interesting, we are hiring. So I'll come to this back later. So I have some disclosures. So I'm a researcher, but my goal is really to help people. And for that, I'm really, really keen to understand what companies need to run clinical trials. And therefore I have a lot of disclosures, and I'm happy to have more. And as my role of the director of the Center for Neurogenetics of the University of Texas, Houston, I'm really motivated to set up a clinical trial platform. So for all pharmaceutical companies interested in executing clinical trials, I'm happy to talk and get the trials going. Okay, so quick disclosure, this is not an educational talk, right? So I'm here to give you a lot of hope that the variant interpretation problem, there are people working on it, and this is not just me, it's like a big community of people here in this session, many funded NIH scientists all over the world, and we're working on this. And I just give you an example how that can look like, that what you receive from a genetic test result, where maybe experts are really uncertain what that actually means and how that can be, used in a clinical trial, for example, as an inclusion or exclusion criterion, or how that might even be a prognostic or diagnostic or be a prognostic marker. People working on this. And I just show you some frameworks around this. And if you're interested in variant interpretation because you're a genetic counselor, physician, we're giving workshops, and the next one will take 3 days where I present probably more than 53 slides in Houston and after the American Epilepsy Society meeting. Okay, so why is variant interpretation important? Because, the most of the variants which you identify in clinical genetic testing, no one knows what to do with them. So, and this is including also for SCN2A. And it's important really to distinguish between the number of variants and the number of submissions. And this is what people typically don't do if they look at these databases. And if you look at the number of submissions, you will realize that even variants that have been seen before and have been multiple times submitted or looked at, people don't know what to do with them still. So, and this is where I have multiple members of my team just focusing on that challenge. So, variant interpretation is a slang. What we actually want to do is variant classification, and just from the variant side. And then, more lately, we are also interested in mechanism. So, and at the end, then it's up to the physician to make the decision how that actually relates to the person you see in front of you. And there you can bring a lot of clinical information about the disease, how that looks. And I will not talk about this today. So that's looking just at the variant, let's say after receiving the genetic test report. So really, you have the report and what can be done. Just a quick example. This is from an ongoing project where we helped Beacon Biosignals, and a project which is unrelated to variant interpretation, but they needed to interpret the variants, and that was presented yesterday. And here we applied a lot of say researchy ways to separate the variants. We look basically, it's really nerdy, the next 5 minutes, you can just believe me that a lot of things are happening. So you can look where the variant is located on the protein, and there's a lot of information from evolution which we can use and which has been shown in the past to be important. We can look where healthy people have the mutation. So even if that variant has never been seen, there are some regions where you don't see mutations in healthy people. We can use these algorithms, which we and others have developed as another layer of, getting a score if that is important. We can look where are actually all the patient mutations, and they are not random, they're not randomly distributed on the NIV 1.2 protein, so the SCN2A gene. You can incorporate this information. You can then do these things just not only in a linear, because you can do this on the molecule, which is the channel which does things. And you see, for example, that the pore region, so in this tube, in this channel, there are more mutations, and you can score this mathematically. And you can look at more databases, which, for example, companies don't have access to, because some of them are very researchy and they're gene specific, and we can access them. So we can even include information from other ion channels. So, for example, other sodium channels, which basically mechanistically work exactly the same. but they do, their job in different cells. But we can use their mutation and their functional results, which has also been tested, and learn a little bit about what's going on. So also how these channels work and where these functional experiments, for example, from Dr. George, have been, tested, and where are they actually localized on the channel, can give also some information to understand about the biology. Long story short, there are some regions which are more likely to cause disease at a regional level than other regions. So we can incorporate this based on data from population, but also from functional experiments. And last but not least, and one of the most important findings, we can just look if the variant has been functionally tested. And there we can make access from the latest data, which has been just, generated through the NIH-funded NNDS Center from L. George. And we can also look at the literature and their efforts to classify everything what has ever been done. And then you can develop a scoring theme, which is aligned towards guidelines from the genetics community. But you can score this a little bit more with all these additional information, and then you get a score for variant pathogenicity. So in this specific project, what was described yesterday, we could basically classify almost all variants into these categories, if they are pathogenic or not. And including those which were in the test report considered as uncertain significance, we could label them. Then we could even, change the classification in some of those in a negative way. So they were reported as pathogenic, but we don't think from our researcher perspective that they are pathogenic. And then we can apply a similar framework for variant interpretation in terms of what we think or could be theoretically for the function. And so here it's really important to also, to score the confidence we have in, is it truly a gain of function or is it truly a loss of function? And it's not binary, right? So first of all, this classification is already a little bit, let's say, crooked. But even within the classification, we have more confidence for certain mutations than for others. And we can generate like a similar scoring scheme. I don't explain now the details why this is important. But in long story short, you don't want to give someone who has a gain of function mutation something which is increasing the gain of function more, right? So we need to understand the disease mechanism. So long story short, we can incorporate also algorithms, what just mentioned, in, as one marker out of many, what the variant is doing. We can look if it's a region, for example, here are functionally tested variants on a sodium channel. And you see that the blue bottom is more likely to be gain of function, whereas the red upper area, the functionally tested variants are more likely to be loss of function. And you can factor in this information. We can also include more research findings because we can look where patient variants are located, and because certain expressions of the disease, certain types of disease which you have due to a mutation are associated with the underlying disease mechanism of the variant, and we can also cluster this on the structure, but I don't want to go too much into detail. So, and again, we can, as the strongest support, take the information if the variant has been functionally tested, and that's literally the strongest information, and use it also as a strong factor in the scoring scheme. Then we can score all, variants which we ever see, and then sort them here into different groups. Okay, so 1 minute. So, and then we can put this into guidelines. And just, I really want to show something researchy, which I've never shown. So, and then but more recently, we have one person who is spending 2 years, Nisha, a very talented postdoc in my team, to do molecular dynamic simulations. So what she's doing, basically, she is taking a mutation and is putting it on the channel. And then she's computing for literally 3 months on a supercomputer and see what that mutation is doing on atom level. And then you get these funny videos here on the top right. And we did this for many patient mutations and then see what's actually happening. And I don't have the time to go into detail, but you can see if you have loss of interactions, or you see if the whole channel is not working anymore. And we can for example, and can see that the agreement with functional tests data and so on. And we have actually quite interesting results for this. And then unfortunately cannot go too much in detail. Okay. So long story short, but I'm here all day. And but this is researchy, and please. So the point I want to make in this very heterogeneous audience, right, is we are working on this, and the path towards clinical trials is not clear-cut in terms of patient selection, but there is a large group of people with very different also opinions, which is good, how to do this, and we are aligning and we're trying to resolve all the issues. Okay, so with that, if any of this is interesting, please talk to me as just about the research, or if you think this is an interesting job area, we are hiring. So thank you very much.
Great. Our next speaker is coming to us live from Glasgow. So there's going to be an echo, a slight delay while we set this up. It was suggested that I should do a dance, but that's not happening. But fortunately, I think Andreas is here, and I'm going to stay away from the microphone.
Can you hear me?
Yes.
I can see and hear you. Can you hear me?
Yep.
Great. Thank you so much for giving me the opportunity to attend to that. I'm really sorry that I can't be there in person, but I really would like to. Can you see my slides? Very good, excellent. This is running very smooth, that's excellent. And I'll try to do all of this in 10 minutes to have some time for discussion. So I'm a child neurologist in Glasgow, and I've been really researching sodium channels for quite a number of years now. And really what I would like to bring to this really is the clinic and how we can use information that we have from other sodium channels to actually help us to predict some of the variants that we find. And the learning objectives really are to describe how in silico prediction tools can aid clinicians to predict the disease phenotype and variant function, and also to understand the limitations of these prediction tools and the value of good clinical characterization of the patient. And if you're interested in this, please have a look at our podcast that's recently been published, where we really talk about this work in a bit more detail. So I really would like to start with just raising your awareness that, of course, there's quite a number of different sodium channels, and we can really learn a lot from other sodium channels. So sodium channels are really important for initiate action potentials in the nervous system, but also in skeletal and cardiac muscle. And you are very, of course, very, very versed with SCN2A, but you have, for example, SCN4A, which is expressed in skeletal muscle. You've got SCN5A, which is expressed in the heart, and then you've got other sodium channels that are expressed in the peripheral nervous system. Yeah, so there's many, many sodium channels, and what's important is that they all share really the same structure. And you will have come across similar cartoons today already, where you've got these 4 transmembrane domains, and you've got the pore loops, and you've got a voltage sensor, and you also have got what's called an activation gate, and that all assembles in the cell membrane to an ion channel where the ions travel through. And let's take this to the clinic. So let's say let's meet Emily. So Emily is a previously 3-month-old child who presents with a cluster of seizures and involving bilateral clonic movements of the upper and lower limbs. And there have been 5 events over a 2-hour period, and each lasting about 40 seconds. And then the parents remarked 1 week earlier they noticed some abnormal facial movements and clonic jerking of the eyelids and lower lip lasting a few seconds. So she's made poor developmental progress since the start of her seizures. There's no family history. And then what you think, and most clinicians would think in a child very young with presenting the seizures, we would like to consider genetic testing. And what you find is that Emily has got a pathogenic SCN2A variant. But of course, what you don't know is at that stage what the future might hold for her. So what we know from the different phenotypes, and you will have had some explanation of this and reminder earlier already, that there's these 4 different groups. One is the benign neonatal infantile seizures that are mostly self-limited. Then the encephalopathies, these are really severe early infantile seizures, less than 3 months of age onset. Then the encephalopathies with later infantile onset after 3 months with infantile epileptic spasm syndrome. And then the 4th group where we have intellectual disability and autism without epilepsy. And thinking about the genetics of this and the gain, whether they gain or loss of function, it's just sort of a reminder of the previous sessions that these are de novo variants. And in the early infantile developmental epileptic encephalopathies, these tend to be gain of function variants. And that means that the channel, for example, cannot close. There might be just too much firing through the channel, and this is then a gain of function. However, if you have the late onset epileptic encephalopathies with autism spectrum disorder, those are loss of function variants, and they're presented slightly later. And for example, the channel cannot open at all, and there's not enough firing through the neuron to the nerve cell, and then you have a loss of function. And when you summarize this and looking, for example, at SCN2A, so if you have those patients presenting with early onset, often before than 3 months, presenting with gain of function, and you would treat patients like that with sodium channel blockers. And if you present after 3 months, you have different seizure types, you have a loss of function, and often you wouldn't want to give sodium channel blockers. So if I now take you back to Emily, who presents at 3 months. So based on the clinical presentation, would you say Emily has a gain of function variant? Would you think she has a loss of function variant? Or would you say you don't know because she's presenting just at the threshold where you don't know what presentation she might have? And this then leads to the question, would you, for example, would you start a sodium channel blocker in Emily at this stage? Would you want to request functional testing prior to starting sodium channel blockers to be absolutely certain of what you're dealing with? And of course, you would like to do that, but of course, time is brain. So the time scale for functional studies is about 6 to 12 months time. There's a significant cost. We have very few laboratories can actually do this worldwide. And clearly there's a mismatch between the high demand and very limited supply. But what we know is that early precision treatment has really the potential to improve quality of life. So this is where the in silico prediction as an alternative to conventional biophysical testing in the laboratory comes into play. And this is a paper by Henrike Heine that we collaborated with, where Henrike looked at predicting the functional effects of missense variants in voltage-gated sodium calcium channels. And she uses basically the disease phenotypes of lots and lots of patients with different voltage-gated sodium calcium channel disorders. And from the disease phenotype, she infers what the function would be. And then uses some machine learning models, and then validated this approach really by with actually functionally tested variants. And she comes up with this map where you can see basically across the cartoon that all those areas that are blue are loss of function, those in red tend to be gain of function. And if you put this, for example, to Emily's variant, you get a prediction of this might be a 62% gain of function. Now, what we've noticed is that, of course, all these sodium channels are really closely related. And you can see here, they're all the different sodium channel genes and the protein sequence. So they're really, really similar. They only differ by a few percentages. And when we looked at different variants and different genes, for example, SCN1, 2, and 8A, we could see that actually the variant location can serve as a surrogate for function. For example, if you have an SCN1A at one particular location, a gain of function has been proved to be gain of function. If you look at SCN8A at the same location, the same variant causes also a gain of function. Yeah, with a different phenotype, but the functional consequences are the same. For example, then if you find at a certain location SCN1A, the loss of function, that gives you another epilepsy called Dravet syndrome. However, if you find at the same mutation SCN2A, you find again a loss of function, but an SCN2A loss of function will gives you an autism spectrum disorder. So we then looked into this and said, why don't we look at all ever published functional tests and experiments on any sodium channel, and we've done that, and really we're able to create a map of sodium channel variants across the entire protein that allow you to actually say at this particular point, we've got this variant, and this variant has been described as having this function. You can see here, all the ones in blue are the gain of function, and all the ones in red are the loss of function, the mixed function. What's interesting is that we found 38 variant pairs, meaning across different sodium channels, who are identical. And what we found is that if you have the identical variant, for example, in SCN, second row SCN4A, you've got a loss of function, which gives you a myotonia. And SCN5A, this same variant also gives you a loss of function and causes you an arrhythmia. Or if you have, for example, in row 5 in SCN2A, you've got a loss of function that prevents visit autism. In SCN5A, at the same position, you also find a loss of function that gives you Brugada syndrome. So if you have the effect of one location, you can actually infer the effect of that particular variant in a different gene. And here we've compiled, with the help of Dennis and Lara in this group, this into an SCN viewer, which basically illustrates all the 430 performed experiments. And then you can look up whether a new variant that you found, whether this has actually already been functionally described in a different sodium channel. And if you do this, for example, for Emily, so you have previously the functional prediction was that this would be 62% gain of function. If you now look at the SCN viewer, what you will find is that there's actually two identical variants at exactly the same position, SCN4A and SCN5A, and these have both been described as being gain of function in experiments. So with this newly acquired knowledge, that take us back to Emily. So based on the clinical and now the new in silico presentation, would you say Emily has a gain of function variant, a loss of function variant, or you don't know? And if you think you've had some indication that this might be gain of function, would you now be more confident to start, for example, sodium channel blockers? And would you still want to request functional testing prior to starting sodium channel blockers, for example? However, if we take this a bit further, then the question was, of course, would you enroll Emily in a gene therapy program? And would you want to request functional testing prior to enrolling her in a gene therapy program? So starting a sodium channel blocker, you can, of course, then stop the sodium channel blocker. But starting a patient on a gene therapy program, that has got different implications because you cannot undo that. So single prediction is not perfect. So we have clearly it is accurate. However, particularly if you have mixed function, that's difficult to predict. Sometimes you can have trafficking differences. This is how the channels travel in and out of the cell membrane that can differ. So the predictions differ as well. And you find that in certain sections of the protein, that's much more difficult to predict than in others. Yeah, and we also know that some variants have been described as gain of function. However, they do not respond to sodium channel blockers, and we don't entirely understand why that's the case and whether this might be due to mixed function. So I think the current tools might be sufficient to inform anti-seizure medication choice, as these tools are accurate enough to, but are they actually accurate enough to inform gene therapy treatments? I don't think they're there yet. And I think that not all patients respond to sodium channel blockers, despite gain of function effects. And we really require better functional maps, really based on experimental data. And I think Al has done a lot of work in this field and contributed to this. And I think that these newly emerging tools, and there are other emerging tools that actually consider the protein structure and effects on function. And what we really need to consider is the clinical and functional data together. That's really, really important. I think that brings me to one of the last points, that we really need to start having really organized epilepsy genetic case discussion meetings where we know that we've got so many more variants and mutations that we find, and they will be classified according to the pathogenicity by our genetics colleagues. However, now we also need to discuss what is the evidence of functional consequences, what can we predict, what is the clinical presentation that is really important. So we really need to have case discussion meetings where we have experts that involves clinicians as well as geneticists and scientists and functional expertise to really come to a conclusion on how to characterize the variant, whether it might be gain of function, and then also make a the consensus decision on which treatment might be the best treatment for that patient. And that's just an overview of these freely available and stable prediction tools that I really highly recommend clinicians to use. And Dennis already showed the SCN portal. And finally, I really would like to thank all our collaborators, particularly Dennis and his team, but also international collaborators in making this work possible. Thank you.